Welcome. In this class we will understand the concept of cell division. Now before starting with understanding the cell division, let's first understand what is cell and how important is the role of cell. So this cell is of a size of 100 micrometers, and within the cell you have so much of processing that you would be understanding today, and that is important for information to be transmitted from the parents to the new cell. So let's say cell cycle I would equate to the cycles or the seasons of a year, and as under different seasons you have the plant that grows, that matures, and finally you have new plants that come up. Similarly is the cell cycle. So under cell cycle we basically classified two primary stages, that is the interphase and the M phase or the mitotic phase. Now this interphase is considered as the longest among the cell cycle. So let's say for human beings we say the cycle is for 24 hours. Of this 24 hours, under interphase you again have three different cycles, that is the G1, S and the G2 phase. And for the human beings, the G1 cycle is of 10 hours, then you have the S cycle which is 9 hours, G2 cycle which is 4 hours, and finally the meteoritic cycle which is simply 1 hour. And this meteoritic phase is the phase of actual cell division. During the interphase, the cell simply grows in size and develops. So this for human beings, let's say, is 24 hours. The same for the case of yeast would be 90 minutes. For the case of bacteria, it would be 20 minutes. So these are some of the theoretical basis or the background that we need to understand before we start with the actual cell division process. Again, when you have one cycle and then you have the next cycle that starts on, the gap between the two is known as the generation time, and that's the time required for one cell to start its process, and it would continue until it divides into two, and when it's dividing into two, it's passing on the complete set of information in both the cells. So you have one cell that divides into two cells. These two cells would further start the cell division and would further divide into two cells. So by this process you have the cell division that goes on, and this is one cycle, this is another cycle. The gap between these two, or the difference between one cycle to the next cycle, would be known as the generation time. Now some of the key contributors in the field of cell division. So Rudolf Virchow explained omnius cellula e cellula, that means cell is derived from another cell. Strasburger explained that nucleus is formed from the pre-existing cells. Again you have Boveri and Fleming who explained the somatic cell division. Finally Fleming coined the term mitosis. Then you have Benedin, Strasburger, Sutton and Winnewater who explained that how chromosome is reduced to half when it goes into the gametes. And finally Farmer and Moore who gave the term meiosis. So you have Fleming and Farmer and Moore who are important. So these are some of the key terms that you must remember while understanding the cell division process. Now in this diagram, in this animation, what we are trying to explain is the basic two divisions that we explained in the starting. So let's say we start with the interphase, and that's the biggest phase. So this would be the M phase, and the rest of all would be the interphase. As we said, in case of human cell division, if we talk about, this would be of 1 hour and the remaining whole cycle would be of 23 hours. So the phase of the interphase, which was previously known as the resting phase, which explained there is no activity that was going, has now been changed to the name of energy phase, because this is the phase during which the growth of the cell takes place, and that helps in the final cell division process during the M phase, that is the mitotic phase. Now this phase is further divided into G1 phase, S phase and G2 phase. So we will understand one by one how these phases vary. So let's first talk about the G1 phase. So first we'll understand the interphase and then we'll move on to the M phase. So during the G1 phase, it's also known as the gap 1 phase or the post-meteotic phase. And what are the basic characters? Because it is occurring after the M phase, so you call it as the post-meteotic phase. Now what are the basic features that happen during the G1 phase? During the G1 phase, you have protein synthesis, you have RNA synthesis, you have accumulation of protein that takes place. Now all this protein, it's important to note, is being made for the purpose so that it could help in DNA replication during the S phase. So during the S phase you have DNA replication. Since all these protein would be helping towards DNA replication, what kind of protein this would be? This protein synthesis would mainly be the histone protein synthesis that would take place in the G1 phase. So once you have the G1 phase that starts, you have the growth of the cell that takes place. Now during the G1 phase, you actually have the growth in terms of size of the cell, but there is very little growth in terms of the nucleus, because the real nucleus work starts with the S phase. So during the G1 phase, you have very little growth of the nucleus, but growth of the cell that takes place during the G1 phase or the gap 1 phase. Now during this phase you have DNA that is 2N in case of diploid and N in case of haploid. However, so this is haploid and this is diploid. Now what would happen during S phase? S phase you have DNA replication or you have DNA that is doubled. So this 2N would become 4N in case of diploid, and in case of haploid N will become 2N. So let's say for human beings we have 2N as 23 pairs of chromosomes or 46 chromosomes. So this 46 chromosome during S phase would become 46 plus 46. So that's 92 chromosomes during the S phase. So S phase is the phase where you have DNA replication that takes place. So your DNA is doubled. It becomes 4N for haploid and 2N for 4N for diploid and 2N for haploid. So that's the basic role of this phase. This phase is also known as the invisible phase. The next to S phase is the G2 phase. During the G2 phase, you have similar activities that happened in the G1 phase. This phase is also known as gap 2 phase or pre-meteotic phase, because after that you have the meteotic process that would start. So we call it pre-meteotic phase. Again, during this phase you would have protein synthesis, RNA synthesis, DNA accumulation, protein accumulation that would take place. However, all these protein synthesis is now for the spindle formation, or we can say for the plasma membrane formation. So there is the difference in the role of protein, which is in the G1 phase and the G2 phase. In G1 it was primarily for the protein synthesis was mainly to help the DNA replication. So the best example of protein here was histone. However, under G2 phase you would have spindle formation that would take place and plasma membrane formation that would take place. So you would have protein synthesis mainly for spindle formation and plasma formation, and then you would move on to the M phase. Now this M phase is further divided into two, that is the chirogenesis and the cytogenesis. So chirogenesis is a phase where you have nucleus that divides, and cytogenesis is a phase where you have the protoplasm that divides or the protoplasm that divides. So during the chirogenesis you have prophase, metaphase, anaphase and telophase, and finally you have the cytoplasmic or the cytogenesis that takes place where you have the cytoplasma division. So within this 1 hour you have ample of cell division that takes place. You have the prophase, metaphase, anaphase, telophase, and finally the cytokinesis, that is the division of the cytoplasma that takes place. Now as the cell divides here, it further goes into the G1 phase for growth. However, certain cell quits at this point and we call this as the G0 phase or the G0 phase. It is also known as the quiescent phase or the quiescent stage. What happens in this quiescent stage? This quiescent stage is known for its maturation and differentiation. So differentiation of cell and maturation of cell takes place during the G0 phase. Normally, once the cell quits into the G0 phase, it won't come back into the cell cycle. For example, the neuron cells, once they mature, they remain in the G0 phase. They do not enter back into the cell cycle. However, in certain cases, for example, the parenchyma cells, you have, once they enter into G0, they come back to the G1 phase and start the process of cell division again. That is routinely not found. Again, as we can see, most of the cell division cycles you have interphase or the growing phase, which is greater than the M phase or the dividing phase. An exception to this is the cancerous cells or the tumor cells, where you have the dividing phase that is greater than the growth phase. So during cancer, in the example of cancer cells or tumor cells, what you would have, the dividing phase, that is M phase, would be much greater than the interphase. However, under all normal cases, you have interphase that is much greater than the M phase or the mitotic phase. Also, it's important to note that this phase is the ultimate phase that governs the further process of meiosis. So let's play this animation. So you have the cell size that is increasing during the G1. It further increases till the time of G2. However, it starts to divide. Excuse me. It starts to divide, and finally you have two cells that are formed from the single cell that started during the G1 phase. Now, to ensure that this cell cycle is smooth, there are three different checkpoints. So we call them as three checkpoints. The first checkpoint exists between the G1 phase and the S phase. It is known as CG1. So you have the first checkpoint, that is CG1, that exists between, or exists somewhere here, and it is a checkpoint between the G1 phase and the S phase. Now what's the role of this checkpoint? If the cell division or the process past this checkpoint, it would move on to the next stages. So what does it check here? It first checks the size of the cell, if the size of the cell is proper or not, the protein accumulation is proper or not, and finally you have the ATP that is synthesized in this G1 phase or not. So these three things are those which are checked under the CG1 checkpoint, that is the G1S checkpoint, the checkpoint that lies between G1 and S phase. The next important checkpoint lies here, that is between the G2 and the M phase, which is also known as the mitotic cycling, that is the CM. We also call it as the mitotic cycling. And during this phase, what does the cell checks out? It checks out whether you have the DNA replication that is completed during the S phase, you have new organelles that are formed during this process of the G2 phase, you have the protein synthesis that has taken place, and if all these conditions meet and you have ATP synthesis, then it moves on to the M phase, and finally you have the third checkpoint that occurs in the M phase, that is also known as the metaphase checkpoint, that occurs in the metaphase stage of the mitotic division. So you also call it as the metaphase checkpoint, and under this you see whether the chromosomes are attached to the spindle fiber. That's the first thing, and secondly you try to check are all the chromosomes aligned on the equatorial plane. When we will talk about the meiotic division, we will see how you have the spindle formations that takes place along the centromere, and you have the breaking up of the chromosomes that takes place during the from the center. If this is not proper, that means there is mutation that would occur. Mutation is the variation from the actual cell division process, and that is one of the kind of deviations from the actual cell division process. So if this is not proper, you won't have a cell that would be divided into exactly two gametes. There could be either irregular number of chromosomes in both the daughter cells. So you would have neither the exact pair of chromosomes, you would have either for 24 pairs of chromosomes or 22 pairs of chromosomes that could be seen, or you would have some changes in the shape or any other form. So that is what is known as mutation. So that occurs or that is checked during the metaphase checkpoint. So there are three important checkpoints: the CG1 checkpoint, the G2M checkpoint and the metaphase checkpoint that occurs in the cell division process. Now why the cell division process is significant or important? It's important because it helps in the multiplication process, it helps to maintain the continuity of the cycle. Now what would happen if this a cell quits at G0 or the cell at G1 is not able to pass into S phase? It would be checked either the cell is not able to pass into the S phase, there is an incomplete information, so it would revert back to the G0 phase, or it would stop here itself and won't proceed further. So cell division could be stopped at any of the points. So what maintains this is the various checkpoints. So again, growth in the cell size, reproduction, shuffling of the genetic traits from mother and father to the daughter or the gamete would be another important feature, and mutations are some of the basic significance of the cell cycle. Now what are the factors that affect this cell cycle? Under factors we basically say there are three primary factors: the size of the cell, the chiroplasmic index, that is the amount of cytoplasm that is present in the cell, and finally you have the mitogens that are present. Now these mitogens are the agents that trigger the cell cycle or the cell division process. Now these mitogens could be different in case of plants and animals. So in plants, let's say you have the cytokinin as a primary mitogen. Under animal cells you have the lymphokinins that are one of the major mitogens. Then you have the epidermal growth factors that are responsible. So these are some of the common mitogens that exist that help in the or that are the basic factors that affect the cell division process. So with this we cover the basic cell division. Now one more important point to know here is what is the difference, the basic key difference between the G1 and the G2 phase. Now when we talk about the difference between the G1 and the G2 phase, first of all, G1 phase is the starting phase, G2 phase is the ending phase. This is known as post-meiotic phase, however this is known as pre-meiotic phase. Now under G1 phase you have increase in the number of chromosomes that occur, no increase in the number of chromosomes because till here you do not have any DNA replication. However, under G2 you have increase in the number of chromosomes that occur. Here you have increase in the cell size and increase or very small increase in the case of cell nucleus. However, in this case you have increase both in terms of cell size and nucleus. Again, you have protein formation or protein synthesis that takes place. Here it is mainly histone protein for DNA replication, which is required for DNA replication. However, here you have mainly protein for spindle formation or plasma membrane formation. So that's again one of the basic key difference between the G1 and the G2 phases. And finally, you have the cell division that takes place, which follows all these basic steps. In the next class, we will be focusing on the various two basic cell division processes, that is the mitosis and the meiosis. We will continue with the section on cell and cell division in few of the further lessons. Till then, have a good day ahead.